So good afternoon, everyone. My name is Victor Henderson. I direct the Stanford Alzheimer's Disease Research Center, and I would like to welcome everyone to our third annual Patient and Research Participant Appreciation Day Educational. That's sort of a long title. Those of you who were listening 2 minutes ago, 1 minute ago, heard Dr. Wyss-Coray, who is our primary organizer, our senior administrator, who's going to keep all of us on track and make sure that everything runs smoothly. I'm really glad to have so many people able to join us today. This has been a momentous year for a number of reasons. And I hope today is sort of a day people can kick back and where we can thank you for what you've done, maybe provide new information on some topics and, engage in a conversation. So the Stanford, well, let me back up, I'm sorry. So this is an appreciation day that involves several different partners. I'm representing the Alzheimer's Disease Research Center, but a really important partner of ours is the Pacific Udall Center. There are a lot of ways in which we overlap, a lot of things that we do in common. The Pacific Udall Center is directed by Dr. Tom Montine, co-directed by our own Dr. Kathleen Poston. We are also supported by Stanford Medicine, and of course, a major part of our support comes from the National Institutes of Health through the Institute on Aging. So the Stanford Alzheimer's Disease Research Center is part of a national network of congressionally mandated centers of excellence supported by the National Institutes of Health. I'm focusing primarily on the ADRC because that's what I represent, and also because I think most of the people join in today, certainly not all, but most of you, or at least many of you, are in some way or another affiliated or helping with our Alzheimer's Disease Center. There are now 31 of us at major universities. We work together to translate research advances into improved diagnosis and care for people with Alzheimer's disease and related disorders. And the ultimate goals are to cure Alzheimer's disease and disorders like Lewy body dementia, like Parkinson's disease, and even more important, to prevent these neurodegenerative disorders from developing in the first place. So our goal is to help people maintain a brain like the one in the middle that's nice and plump and healthy, and not to develop a brain that looks a little bit more withered and affected by disease. I'm really very happy this year. 2020 has not been a happy year in some ways, but it's been very good in the sense that the Stanford ADRC received 5-year renewal funding from the National Institutes of Health. This is an important milestone for us. The center was reorganized in small ways, the way other centers are being reorganized during the next cycle of NIH funding. But most of the faces here are familiar. We've been fortunate to be joined by new faculty, new staff, and I wish I had time to introduce you to those people now. I want to mention particularly our Associate Director, Dr. Kati Andreasson, from whom you'll be hearing more in a few minutes, but she is the center co-director. I also very much want to thank our volunteers within the Alzheimer's Disease Research Center. There are 423 of you who have volunteered to make this center successful. I think our NIH reviewers acknowledge this and were impressed by this. Virtually every one of you have volunteered not only to come in and to be examined in person and to undergo memory and other cognitive tests, but almost everybody has provided a blood sample for research and has consented to longitudinal follow-up over time and eventually to brain donation. And very many of you, in some instances, most of you have also provided other resources to help our investigators. And these include things like a small skin biopsy from which skin cells can be grown, fibroblasts, which then can be converted by other researchers into neurons for their research to help find answers to the disorders that we're concerned with. We also have been collecting stool samples to look at the microbiome, which we think is really important in both health and disease. And for many of you, you have given generously spinal fluid from which a lot of different studies can be done. Most of you have volunteered to have brain scans done, either an MRI brain scan or PET scan, or in some instances, both of these. So thank you so much for making this possible. I think at this point, I'm going to turn things over to Nusha, who will introduce the program a little bit, tell you some housekeeping rules, and then I'll return after that.
Thank you, Dr. Henderson. Welcome, everyone. On behalf of myself and Kristina and all of the RAs who've been so kind to help us out, Veronica Ramirez, Talisa Meadowcroft, Nicole Corso, Michelle Collado, and Maria Lucia Campos. We welcome you, and we greatly appreciate your continued enrollment and participation in the center. In terms of some housekeeping, the order of the speakers will be Dr. Henderson, followed by Dr. Andreasson, followed by Dr. Wyss-Coray, and then we'll have an open discussion forum, a break, and then Kristin Morris and Lauren Stroshane will be the second session with another opportunity for question and answer. Chat button, again, is reserved for any technical issues, and a few of the RAs are helping with that. After each speaker, we've allowed a little 5-minute cushion in case there's a very pressing or clarifying question, one or two of those. The first session will be moderated by Nicole Corso, the second session by Talisa Meadowcroft, but primarily we'll be taking questions and answers during that open forum, which the first one will be approximately around 2:20 p.m. for about 15-20 minutes, and the second one will be around 3:40 p.m. After Lauren's speaks, and then the first session will be after Dr. Wyss-Coray speaks. So the way we'll do this is if you could please cluster questions, we're going to cluster the questions per speaker. If you can put the speaker's name or ideally just their initials. So for Dr. Henderson, it would be VH, Dr. Andreasson, KA, TWC, Dr. Wyss-Coray, KM for Kristin Morris, and LS. That way we can kind of go through the questions quickly. You can type them. If you would really like to speak your question, there's a raise hand button on the bottom of your screen. Please raise your hand. You'll be enabled to unmute. It'll ask you to unmute, and then you can ask your question. I think that pretty much does it. And we can continue with the rest of the, so please type your questions in the Q&A. That's the main thing I wanted to make sure that I specified. Thank you. Dr. Henderson, sorry.
So I'm trying to see if to get back. Are we good with the screen sharing?
Almost, there we go, yes.
Great. So thank you, Nusha, and thank you, participants. I'm going to be speaking for a few minutes about mild cognitive impairment. This is something I think most people know a little bit about, and what I want to do is to go over the concept a little bit and to speak a little bit about why I think it's important. So mild cognitive impairment, as one might expect, is sort of an intermediate stage between normal aging and dementia. There's something in between, and the term that's been developed for that is mild cognitive impairment. So one can think of perhaps a continuum, or at least a projection going from normal aging to mild cognitive impairment to dementia for people who are destined to go along the spectrum. Certainly not everyone does, and it's our hope to, in the future, have a way to ensure that nobody goes along this pathway. But this is the pathway that we're speaking of. So normal aging, I view this as an age-associated change in the brain, changes that do not in and of themselves cause dementia. Some of these are physiologic, sort of built into the brain. Some of these are related to the wear and tear of aging, but these are changes that by themselves, even over time, don't build up enough to cause dementia. So abnormal brain aging then would be age-associated changes in the brain that can, by themselves, end up with dementia. And in this view, then, mild cognitive impairment would represent an earlier change in the stage of abnormal brain aging, where there are some cognitive, memory, reasoning difficulties, but not reaching the threshold for dementia. So it's along the continuum with dementia. As we think about normal brain aging, normal changes with aging, there are different kinds of knowledge and skills. Factor psychologists for a long time have spoken about the kinds of abilities that draw largely on acquired skills, knowledge, and experience. They use the term crystallized intelligence to this. It's sort of what you know and how to do certain things. And there's a different group of abilities based more on learning new information, abstract reasoning, problem solving, and the term that is used for this sometimes is fluid intelligence. The National Institutes of Health has developed toolboxes to help researchers, including cognitive toolboxes, and they have a battery of tests that they use to look at crystallized intelligence and fluid intelligence. And in the NIH norms, looking at sort of a composite of crystallized intelligence, looking at different age groups shown along the axis in the bottom, from a fairly young 18 to 29-year-old group to an older age group. Over time, these kinds of abilities stay about the same for both men and for women. In contrast, for the kinds of abilities that we're referring to as fluid intelligence, there's a decline over age. The decline is seen for both men and women, and it's not a decline that starts at age 65 or age 70 or age 50. It's a change that starts even in one's 20s. And this is pretty consistent in other kinds of batteries as well. And so there's some kinds of skills that are maintained throughout the lifespan up until maybe ripe old age, and other kinds of skills that aren't maintained in the same way. So in terms of abnormal brain aging, I think now pathologists have a fairly good handle on what this is. And these are sort of the usual suspects. These are the changes in the brain that underlie Alzheimer's disease, vascular dementia, Lewy body dementia, the group of frontotemporal dementias, and there's some other specific causes, Huntington's disease, for example, and several other things. But by and large, pathologists looking at the brain can point out things that have contributed to this abnormal brain aging. So this is a slide that represents the plaques and the tangles of Alzheimer's disease. This is an MRI brain scan that would show evidence for vascular dementia, white matter change here, a stroke shown here. This is sort of the frontal and temporal lobe shrinkage that goes along with some of the frontal temporal dementias. This is a Lewy body up here that would go along with Lewy body dementia and also with Parkinson's disease. And so these are well known to neuropathologists. So when we think about mild cognitive impairment, and we think about the two models of aging and dementia, the first model is sort of a gradual continuum with sort of these changes build up in everyone. And the other is where normal aging is conceptually different from mild cognitive impairment. And it's really the second model that I think is the right model, at least as a first approximation. So thinking that there's something between normal and frank dementia is not a new concept. It's something that's been known and discussed for a long time. A generation ago, people were talking about benign senescent forgetfulness or age-associated memory impairment. And it wasn't until 1999 that the concept of mild cognitive impairment was developed. And there have been some other recent concepts that have come after that. But it's really this MCI, this mild cognitive impairment, that is caught hold. These different terminologies here, these different terms, don't all refer to exactly the same thing. The definitions vary a little bit, but it's sort of this intermediate ground between dementia and normal aging. The original definition was based on memory alone, and the reason for that is a lot of dementia, perhaps most dementia, is due to Alzheimer's disease. And for most people with Alzheimer's disease, the earliest manifestation is memory. So the original definition was based on someone noticing, or someone's family member noticing that there was something wrong with memory, people were still able to perform normal daily tasks without difficulty. General cognition was generally taken to be average, sometimes defined by neuropsychological test scores, but there was an abnormal memory for age. And if this was based on test scores, it was usually defined as being 1.5 standard deviations below sort of what the expected memory scores were. And this translates into the bottom 7% of the population. And of course, there was no dementia, but no dementia means essentially that people were able to handle their activities of daily living in a normal fashion. We now recognize, we clinicians, we researchers, 4 kinds of mild cognitive impairment. It includes the just memory form that I showed in the last slide that was developed in 1999. But we also know that for some people, memory is not the first thing that occurs, certainly in disorders other than Alzheimer's disease. Spatial problems or planning and reasoning problems or speech and language problems can occur early. And even in Alzheimer's disease, sometimes this can be the case. And so the 4 categories include memory by itself, memory with something else. If memory is spared, then 1 or 2 other kinds of cognitive domains being affected. But the underlying principles for defining these based on neuropsychological test scores and the ability to conduct daily affairs normally or adequately, that part hasn't changed. How common is mild cognitive impairment? I don't think it will surprise anyone here that is more common with age. This is a publication a couple of years ago looking at 30 different studies of mild cognitive impairment. The way the definitions are interpreted varies a little bit from study to study, but in essence, you can see in 160s, of the left-hand portion of the slide, the number of people who've met various definitions or criteria for mild cognitive impairment is pretty low. And it increases to about 25% in the 9th decade of life. And this would go along with the known increase in the rate of dementia with increasing age. Now, what underlies mild cognitive impairment? This pie graph comes from one of our sister ADRCs. This is at Rush in Chicago. And they had followed a group of people with, I think, annual cognitive testing or every other year cognitive testing to the time of death. This was a healthy group. The average age of death was 87 years. And some of the people have been adjudicated as being normal cognitively, some as having dementia, and some as having mild cognitive impairment. And when they look at the brains at the time of death, over half of them had Alzheimer's disease changes, the plaques and tangles that I talked about before, not necessarily by itself, but Alzheimer's disease by itself or with something else. Another proportion had strokes, or they had changes of Lewy bodies, and another quarter had none of these changes, none of these 3 changes, although there might have been some other changes present. If you look in the same group, people who clinically were thought to have dementia and Alzheimer's disease, not surprisingly, most of them did have Alzheimer's pathology. Not all of them, but most of them did. Some of them had other changes, Lewy body and stroke, and a few of them had different changes, but not these 3. For normal, moving over to the right-hand part of the slide, it was a little bit surprising. People who are cognitively normal sometimes have these changes as well. Some of them did have some plaques and tangles. Some of them did have some vascular changes, strokes, or Lewy body changes, and a lot of them had none of these changes. So in this sense, the model between normal aging, mild cognitive impairment, and dementia might be more the model 1, where there's sort of a blurring rather than the distinction that I tried to make earlier. But I think conceptually that distinction still holds. I still think model 2 is the right way to look at this. So this is a slide I thought I'd take it out, but I didn't, so I will talk about it. One of the important findings in Alzheimer's disease, well, let me back up. cognition in the brain, of course, is related to the nerve cells, the neurons within the brain. And there are about 86 billion neurons in the brain. And nerve cells communicate with each other by means of small connections referred to as synapses. And there are a lot of these. There are about 100 to 1,000 trillion synapses in the brain, the human brain, the adult human brain. And that's a number with a lot of zeros. If we look at the plaques and tangles of Alzheimer's disease, and we correlate this with disease severity, the plaque burden correlates with whether someone has dementia or not, but it's not high correlation. If you look at the number of tangles within nerve cells, then that correlates better with the degree of dementia or developing dementia. But if you look at the number of synapses, then that correlates even better. That's really the best marker of how impaired someone is. So looking at the bottom left of the slide, this shows a healthy looking pyramidal nerve cell within the cortex. A lot of branches bringing information into the nerve cell, sending information out. And in each of these branches, there are multitudes of these synaptic connections. And during the course of Alzheimer's disease, before there's a dead nerve cell shown on the right here, before the neurofibrillary tangle has sort of overtaken the nerve cell, there's a massive loss of these sprouts, of these dendrites. And going along with that, there's a massive loss of synapses. The lower right-hand portion of the slide shows markers of synapses within the brain. These little green dots each represent a synapse. The black holes are the nerve cell bodies sort of cut through, so you're missing the outside of the nerve cell. And in Alzheimer's disease, you can see that the number of neurons, the number of synapses is massively reduced. So there are evolving concepts of mild cognitive impairment in Alzheimer's disease. This is primarily being used in a research setting, but I think a lot of things in research bleed over into the clinical setting, and it's important to recognize and understand this. So researchers now speak about mild cognitive impairment due to Alzheimer's disease. So this isn't Alzheimer's disease in the way we use it clinically to mean dementia. This is Alzheimer's disease, meaning that there are some plaques and tangles that the researchers think are causing the mild cognitive impairment. They're also speaking about Alzheimer's disease in people with no memory loss, no cognitive impairment. So this would be preclinical or asymptomatic Alzheimer's disease. I worry about this use of terminology, but this is what's going on in the field right now. So mild cognitive impairment due to Alzheimer's disease would consider a number of things. But for both that and preclinical Alzheimer's disease, the important things are genetic testing, which is important in rare circumstances, but more importantly are biomarkers. Biomarkers are biological correlates. They aren't diseases in and of themselves. And the biomarkers that are most important for Alzheimer's disease are biomarkers related to amyloid, to tau, and these can be measured most easily right now, at least, within the spinal fluid or in certain kinds of PET scans. So coming back to a typical microscopic section of the brain of Alzheimer's disease, there are these large neuritic plaques between nerve cells. There are these tangles within nerve cells shown here. And the biochemistry is such that in the center of most plaques with different stains, there would be an amyloid core, which is an abnormal accumulation. And within most tangles, it would be shown that these are made up of a form of tau protein, a normal protein, which has been modified and misfolded in a certain configuration. So a lot of biomarkers focus on amyloid and tau. These can be measured from a spinal tap, a lumbar puncture, for example. And I thank again all of you in the ADRC who have consented to spinal taps, because there are a lot of different studies that can be done with the CSF in addition to looking at sort of these classic biomarkers. And the other way is looking at amyloid or tau in the brain. Dr. Morminos lent me these slides showing First, at the top here, an amyloid PET scan result from people within our Alzheimer's Disease Center. The first shows no amyloid out the cortex. This is sort of a non-specific white lighting up in the middle for people, which doesn't represent amyloid. But for some people with mild cognitive impairment, there will be amyloid that does show up in the cortex here and here. Not all, certainly not all, but for some there will be. And for Alzheimer's disease, of course, we expect this pretty routinely. The other more experimental and newer tracer is related to that other protein, tau. In normal older people, there's usually no tau at all, or maybe a little bit down within a part of the temporal lobe, but usually none. But for mild cognitive impairment, sometimes tau will show up within the temporal lobe, within the other parts of the cortex shown up here. And in Alzheimer's disease, a lot more. And so these are markers, not diseases, but these are biochemical markers that sort of give us clues as to the underlying pathology. A lot of drug trials are now looking at biomarkers and targeting people with mild cognitive impairment, particularly drug trials that are targeting amyloid within the brain. And to give one recent example, and one example that has been in the recent news and may continue to be in the news for the next, well, we don't know for how long, but, and that is aducanumab. That's a trial that's being conducted or was conducted in people with mild cognitive impairment or mild dementia due to Alzheimer's disease. It's a drug that's given by intravenous infusion. It's a drug that does a good job of removing amyloid from the brain. And the right-hand part of this slide shows people at baseline who had a lot of amyloid. Some received a placebo drug, some received lower and higher doses of the experimental treatment. And amyloid was indeed removed from the brain. There were some side effects, but in general, this could be done. More importantly, though, is after a year and a half, the drug showed only a very small benefit in one very large trial. And in a second very large trial, that benefit wasn't there. Alzheimer's disease, rightfully, I think, has been defined by the FDA as an unmet need, meaning that the FDA can approve drugs without the stronger proof it needs for other kinds of diseases. The Neuroscience panel has recommended approval of this medication, but the advisory panel, which is the next step in the FDA process, has thought that the evidence is too weak and we don't know what the final FDA ruling will be. So the take-home messages are then, there's a conceptual distinction between cognitive aging and mild cognitive impairment and dementia, the latter two being abnormal forms of aging, that some cognitive abilities hold up well with aging. We refer to these abilities, sort of knowledge, as wisdom, but other abilities are more affected normally, we think. The biochemical changes, the plaques and tangle changes of Alzheimer's disease can occur years before symptoms appear, even before MC appears. The term Alzheimer's disease is now being applied in research settings to people with mild cognitive impairment, particularly when there's some biomarker evidence of amyloid and tau. And I would point out that there are really no effective treatments for MCI related to Alzheimer's disease or MCI related to Lewy body dementia, but it's a really important area on target for research. And we think it's particularly important within our ADRC because knowing who has some of these changes and doesn't go on to develop dementia or does it slowly, and who, understanding who is resilient to these changes, who develops these biomarkers in the first place, despite, in some instances, a genetic predisposition, and who doesn't. I think there are really important things to be learned there. That's one of the things we hope our center will contribute to, and one of the things I am particularly grateful to all of you for sort of helping us with. And so with that, let me thank you for your attention. Thank you for giving up your Saturday afternoon to be with us. And I think here, looking at time, I'll wait for Nusha to tell me otherwise. I think we might save questions to the Q&A period, Nusha, unless you think we should take one or two now. I'm not really looking at the chat box on my end, and so I will defer to you in this.
Yeah, I don't see anything that's come up. So I do know that Dr. Andreasson is pressed for time. So I want to be respectful of her time, and perhaps we can just move right to Dr. Katya Andreasson's talk.
We shall do that. And a really brief introduction. As I said before, Dr. Andreasson is the Associate Director of our center. She is a professor in the Department of Neurology at Stanford. She's vice chair in charge of research. She is a neurologist with a basic science interest in some of the underlying mechanisms related to these neurodegenerative disorders, and will be telling us about the role that inflammation seems to play in these disorders, including Alzheimer's disease. So Dr. Andreasson, thank you for joining us.
Yes, thank you. Thank you, everybody, for coming this afternoon. It's really nice to be with you. It's a really good day. um, I'm going to shift gears a little bit. Dr. Henderson has been made a beautiful overview of sort of the pathology and the clinical characteristics of MCI and Alzheimer's disease, and then leading up to the most recent trial with the antibody to amyloid. So I'm going to switch gears a little bit and talk a little bit about inflammation, and we're going to sort of shift more towards a more basic science biology perspective. And so just for those who need a refresher from biology. So the immune system really is very complex. We have an immune system to fend off infections, for example, bacterial or viral. We also have an immune system to fend off internal pathogens, for example, cancer. And it's important to know that the immune system is not simply just a couple of cells in one particular place. It's a very disseminated, sort of decentralized kind of organ system. And here, I think you can see my cursor here. The immune cells actually come from the bone marrow, and they populate a number of these other organs that are shown here. And I think what's more familiar probably to many of you is when you get your bloods drawn and you get a CBC, they do measure a number of these immune cells, and they're actually quite important. I'm going to end up talking about monocytes, not so much neutrophils and basophils, but these are part of the innate immune system that, as I will show you, actually play a very important part in age-associated diseases, in particular Alzheimer's disease, and probably other forms of dementia as well. So in the blood, we have all these circulating immune cells, and to get a bit more granular look at what they are, there are two branches of the immune system, and the one branch that is particularly relevant to Alzheimer's disease and other age-related diseases is what we call the innate immunity or innate immune response. And these are sort of the rapid responders. So for example, if you get a bacterial infection or a virus, there's a lot of COVID out there. The rapid responders here are the macrophages and these cells over here. And the adaptive immune response is also probably important in age-dependent or age-associated diseases like Alzheimer's, although there's much less known about it. And I want to draw your attention to the macrophages, which is these are the real workhorses. And if you think about macrophages, they are sort of the sentinels. They are always patrolling through the blood, through every single organ in your body, your liver, your lung, and your brain. And in the brain, the macrophages have a different name. They're called microglia. And they have an entirely unique biology and responsiveness. And this is kind of what we are studying in my lab. We're looking at how the innate immune system, how it changes with aging, what goes wrong, and what are the pathways that we can identify that are not working quite as well and that we potentially could target to sort of allay the severity of this progression of disease. So this is kind of a new concept, probably over the last 10, 15 years. And I have to say that the dementia field has come to it somewhat late. It's only more recently that we are indeed acknowledging that there is a very important, probably causal a relationship between inflammation or disrupted inflammation, or what we like to call maladaptive inflammation, and the development of various age-related diseases. And here, this is a very nice review where you can see that there's a whole variety of different disease states that are definitely enhanced, if not possibly caused, by an abnormal or a deteriorating immune system, and in particular, a deteriorating innate immune system. So these would be the macrophages, the monocytes, and in the brain, it would be the microglia. So for example, cardiovascular disease, this is a big one. So if any of you who are taking statins, that's because you need to lower your cholesterol. And turns out that atherosclerosis, which is where the macrophages of the blood in the blood vessels, in the arteries in particular, they become filled with cholesterol, and they cannot actually break it down, and they become very, very inflammatory, which then sets up this feed-forward loop where you get even more inflammation, and that leads to thrombosis. The platelets will come and stick, and suddenly you have a clot. So this is that's an inflammatory cause. And so there's some other cancer, very important in somewhat an interesting way, where cancer cells can fool the immune system, and so that the immune system doesn't even know that the cancer cells are there. And those of you who are familiar with these very dramatic advances in cancer therapy or immunotherapy, where they have developed antibodies to target these receptors that kind of fool the immune system into ignoring the cancer. Frailty, very important. Arthritis, loss of cartilage. These are all very connected to maladaptive inflammation. Metabolic syndrome and obesity, there's a tremendous amount of inflammation that comes out of these conditions. So it goes on that, and a lot of these diseases, as you can see, are also increasing as we get older. So what we conclude is that there's something that's happening with aging. What my lab is very interested in understanding is how the innate immune system changes with age. What are the major factors that sort of make it decline so that one becomes more predisposed to these types of age-related diseases, including Alzheimer's disease. And so the characteristics for these first responders, microglia, macrophages, monocytes, is that they don't clear toxic debris. So they don't do very well on clearing a bacterial infection, which is why bacterial infections are so much more severe and serious in older ages than they are in younger. They don't turn off the initial inflammatory response. And the initial inflammatory response consists of a lot of immune factors that really rev up the immune system, leading to generation of various substances that can be quite toxic, like free radicals, or something called a protease, which is a protein that clips and chops off proteins. And then most importantly, you lose this sort of return to youthful homeostasis. So when one is very young, you can pretty much, take any kind of hit and you'll be fine. But it's different as you get older, and the immune system is no different. And then when we get into the brain, which is a very different environment, because there, not only do you have your microglia and blood vessels, but you also have neurons, you have circuits that are communicating with each other. So these are very exquisitely sensitive to disruptions in the environment. And one of the major roles of microglia in the brain is to patrol and to always be moving around, making sure that the environment there is maintained in this sort of pristine fashion to allow neurons and synapses and circuits to work properly. And so microglia are very important in maintaining this homeostatic environment. You have heard from Dr. Henderson about amyloid. So they are extremely important in getting rid of this amyloid that you see in these MRI, in these PET scans. And they're also very important in maintaining and pruning synapses that don't work, sort of sculpting different circuits. So they perform a very physiologic role as well. So you can imagine that as we get older, this phenotype actually really changes. So here I have a little depiction of a microglial cell. Here are the peripheral macrophages and monocytes that I was describing. And so with aging, what happens is you go from this very youthful, healthy, you can take everything phenotype to a much more fragile and less effective phenotype where they don't, this is a term phagocytosis, which is extremely important function where you can where these cells gobble up toxic debris or toxic proteins or bacteria, then they become very pro-inflammatory. So a lot of fairly nefarious immune factors that are detrimental. So when we think about Alzheimer's disease, and here on the x-axis is age, and this is sort of the curve, and the principal risk factor for Alzheimer's disease is age, and so it does go up with aging, and but what we would like to do is prevent it, obviously, and that's what my lab and others around the world are very interested in doing. And what we want to do is we want to prevent it early. So for example, at the mild cognitive impairment stage that Dr. Henderson was talking about, or even before, actually, because, and what we're desperately searching for now are biomarkers. So sort of specific factors that start to change even before we get the clinical diagnosis of MCI. And so the obvious, the wonderful outcome here would be if we could find such a biomarker, and if we had a therapeutic that would act early enough, we could really reduce the incidence, the prevalence, which is here on the y-axis, quite significantly. And the NIA and the National Institutes of Health and other organizations are sort of stipulate that if you can simply delay Alzheimer's by onset by 5 years, you would reduce the prevalence by 50%, which is quite a wonderful objective. So Anyway, oops, here we go. So here, this is somewhat technical, but this is how in the last decade now, the field has come around to recognizing an immune cause of Alzheimer's disease. And these are studies, and this one is actually from 2013. There have been some updates since then, but this was the major study where they combined a lot of human genetic studies. So these are studies with thousands of human subjects in various ADRCs and other consortia. And what they did there is they sequenced their DNA and looked for differences between DNA and correlated that with whether or not someone was had cognitive decline that was consistent with Alzheimer's disease. And what was really remarkable was these dots here. Here is a significance level, and all these dots here represent genes. And there are quite a few dots that they found in this new, very, very large meta-analysis. But what you notice, I've circled a lot of these in green, and it turns out that most of these are immune genes, and they're not just immune genes, they are innate immune genes. So this means that these genes, if you have a little bit of a variant, you have a slight increase in risk of getting Alzheimer's disease. And of course, maybe some of you will recognize ApoE up here. This is a very, very important risk factor. It doesn't necessarily cause it, but it enhances the risk. So there's a slight sort of nuance here. So this was really game-changing, I think, in the field. And I had been previously working in this area, and I could not get my grants funded because nobody was thinking about inflammation. But now suddenly that has completely changed, and it's very gratifying. But anyway, that aside, what I wanted to connect the dots here between what Dr. Henderson was telling you about the pathology, which involves amyloid, and then the second pathology, which I think you referred to, which is tau. These are, oh, excuse me. Gotta mute this. There are two things that will activate or that will wake up a microglial cell in the brain. So both of these proteins, here on top is a diagram of how amyloid is actually made in the brain. And here is a diagram of what happens to this other very important protein that misfolds called tau. And both of these, amyloid accumulation is necessary to get Alzheimer's, but it's not sufficient. So you can have amyloid in your brain, but not necessarily have any cognitive decline. But it's when you start to get the tau involved, that's when things kind of get dicey. And what's very important is that both of these misfolded proteins, the A beta, the amyloid beta fibrils, as well as the tau aggregates, are very powerful activators of microglia in the brain. And so you set off this real response, which can be quite maladaptive within the brain, where now the microglia are responding to these two misfolded proteins. They launch a maladaptive immune response. They spew out free radicals, proteases, immune factors that then can damage neurons and synapses, and also basically feed forward or amplify the immune response. So what we want to do is really focus on how we can quiet down these microglia and basically make them function better so that they can gobble up this stuff instead of becoming hyperactivated. So early on, and this was many, many years ago now, 2008 and before, there were these very large observational studies. This one by Vlad et al. Is actually out of the VA, and there are a couple of other studies that have since, that were actually performed beforehand. And what happened here was there was subjects were taking non-steroidal anti-inflammatory drugs, which I'm sure many of you have taken, indomethacin, naproxen, ibuprofen, and these are sort of classic anti-inflammatory medications that we can get over the counter for aches and pains. And it turns out that just taking these here and there, and that this was the first study by Stewart et al., just taking an NSAID here and there was associated with a reduction in future risk of getting Alzheimer's disease. And this was followed by a number of other studies, and then culminating in this one at the VA, where it looked like this is 100% risk, but you can see how your risk declines the longer you take NSAIDs. And now at this point, about 4 to 5 years into taking NSAIDs, they start to see a real drop in risk of getting Alzheimer's disease. Now, this was all very encouraging, very nice. I would definitely not advise you to go and take NSAIDs because NSAIDs have a lot of other side effects. For example, kidney toxicity, and GI toxicity. So NSAIDs are not the answer. However, what's really interesting is that NSAIDs block this set of enzymes here at the tippy top of this pathway, which involves arachidonic acid, which is a very common or a very abundant membrane lipid in the brain. And it's metabolized by the cyclooxygenases, or COX-1 and COX-2, to this precursor here, which is called PGH2. And I just want you to remember this one because it's actually a very important precursor. It's a precursor to the prostaglandins, which is what I'm going to talk about now. And the PGH2 gives rise to 5 different prostaglandins. One of them is this one, PGE2. And this is a very nefarious, inflammatory molecule. And it will bind to a series of receptors. So this is like a key and a lock kind of concept. So you have a receptor sitting here on an immune cell or a microglial cell. The prostaglandin E2 binds to it and activates it. And so we have actually been looking at this pathway because we were really intrigued to figure out how NSAIDs worked, and knowing full well that it's absolutely not a good idea to take NSAIDs to prevent Alzheimer's because there are just too many potential side effects. By taking NSAIDs, you block not just this production of this, but you block production of beneficial prostaglandins. So that's why it's not a good idea to take NSAIDs. So, and then Tom Montine, who is here at Stanford, long time ago when he was at University of Washington, he and his lab and colleagues did a very nice study looking at spinal fluid in human subjects. I think these were people who had enrolled in the University of Washington ADRC, I think. And what he did is he measured the levels of these different prostaglandins. You can see here, here is a set of control subjects, and here are the subjects with Alzheimer's disease. And you can see how high that prostaglandin E2 is. The other prostaglandins, I was mentioning that there's some good prostaglandins, and this one in particular, where is it? This one is very, very good, and it's decreased in Alzheimer's disease. So you have this kind of double hit where you have too much of a bad prostaglandin and too little of a good prostaglandin. So we have been looking at this pathway for quite some time now, and what we have discovered, and this has been over the last 15 years, we've been chipping away at this, but we have discovered that this particular receptor is really the mother lode of bad outcomes. So this receptor, when you make PGE2 and it binds to the EP2 receptor, this suppresses all the good functions that a microglial cell or a macrophage needs to kind of keep you healthy. So for example, it suppresses phagocytosis, which is gobbling up of amyloid, for example. It suppresses turning off the inflammatory response. So that means that these, if when EP2 is activated, you just keep getting more and more and more inflammation, and it never turns off. And there's a couple of other very important pathways that are important for maintenance of synapses and circuits, and just the creation of a very beneficial homeostatic environment within the brain for the neurons and the synapses to work properly. So what we're really doing, and I'd be happy to talk offline about what we're doing a lot to look at this pathway and to look at what is downstream of this. And we have a number of very promising pathways. And our ultimate objective is to, first of all, prove that this will be a very effective target to inhibit in people. We've got a ways to go there. But also to look at other targets that are associated with this pathway. And we have a couple of additional pathways that we're looking into. And so our sort of goal and our hope is that if we can, this is what we'd like our microglia and myeloid cells to look like, very healthy. Unfortunately, with age and with amyloid accumulation, they really don't do very well. They become more pro-inflammatory. They lose all their essential homeostatic functions. And what we'd like to do is now just kind of turn back the clock. And we have now identified at least 2 different pathways where we can do that. That's where we are. And anyway, thank you very much for your attention. And it's really fun to share this with you. Thank you.
I think, Nicole, you were going to take maybe 1 or 2 questions for Kati. Maybe read them out loud so she can answer them live.
Sure. So the first question we got was, is there any effect of sugar in inflammation and neurodegenerative disease, and could exercise potentially neutralize the effect of sugar?
Yeah, so there is a real association with elevated sugar or glucose intolerance, diabetes, metabolic syndrome, with heightened levels of inflammation. And I'm not aware, I am aware that there's a lot of research in exercise. I can't quote the articles to you. I haven't really had a chance to read through them, but there is increasing evidence that exercise is highly beneficial. And this is actually very well established. And it's one of the things that we are always recommending to our patients is, even if you can't do any exercise, do something, do a little bit and work your way up, because it's very, very beneficial. It's intrinsically anti-inflammatory, plus it helps with sugar control.
And I might add there, if I might, that we'll talk more about exercise, I think, in the Q&A break or session that's coming up a little bit later.
Let's see.
Nusha, is there time for any additional questions? There was a couple more.
Kati has to leave, so I think if we can, Tony, if you could kindly bear with us, let's take care of Kati Andreasson's questions now, and then we'll take the other questions during the Q&A. Thank you.
So we had, oh, go ahead.
No, please go ahead.
Okay. So we had a couple of additional questions kind of related to the question that we just asked, but is there any specific role of diet in suppressing anti-inflammatory effects? Any like foods or supplements that are directly related to that?
Yes, so there's a very large effort by the National Institutes of Aging, and actually Dr. Henderson probably has a lot more insight into this in terms of the trials. The Mediterranean diet is turning out to be very, very anti-inflammatory and very good for cognition. So a combination of exercise and diet is really, in terms of holistic approaches, is extremely important.
And then another question we had was one of our participants had a brain scan done through Stanford through a different study, and it was looking at inflammation. They believe the radioactive isotope that was used was carbon-11, and they were wondering if you could speak to that and what that study was potentially looking for with that isotope.
Yeah, so I'm not too familiar with the various isotopes, but this was probably a TSPO PET scan for inflammation. The TSPO is currently the only PET scan agent for inflammation. However, that doesn't mean it's the best, and it definitely can, it detects inflammation, but it also detects other things. And so it's not super, super specific for inflammation. And that's one of the things that we're quite interested in, is at the same time we want to, I was mentioning biomarkers, which is very, very important. And if we could find a biomarker that sort of changes just before we get MCI, that would be very valuable. And if we have inflammation already rip-roaring in the brain at that time, wouldn't it be nice to detect that very specifically? My sense, I don't know if TSPO is sensitive enough to do that at this point, but it's the best we have. So it was reasonable to have that done.
Nicole, I think we need to sort of pay attention to time. Is there one final question? And
Those
then,
were all the questions that we had.
great, thanks.
Mm-hmm.
Well, thank you very much.
Thank you.
So thank you, Dr. Andreasson. And let me introduce our next speaker, my colleague, Dr. Tony Wyss-Coray. He has been an important leader within the Alzheimer's disease research since its inception. He leads the biomarker core in the new renewal of the Alzheimer's disease Research Center. He is a professor of neurology. He has an endowed chair. He trained as an immunologist and has been a really productive, basic laboratory investigator looking at a number of different factors. And he will be speaking today about one of the studies that he's investigating, looking at young blood for old brains. So Dr. Wyss-Coray.
Thank you, Victor. Can you all see my slide and hear me?
I see you. I don't see your slide right now.
Okay. Let's try this again.
Looking more promising. Looking, yes,
Okay, how's that?
perfect.
Okay.
Good.
Good afternoon, everybody. And thank you so much for participating, for tuning in today, but in general, for participating in our studies of Alzheimer's disease and related dementia. Your participation is really crucial for us to understand this disease. If we look at this image here of a parking lot, I think many of us have sort of this experience of parking a car in a busy lot and going for an hour or so of shopping and then coming back out and don't quite remember where we parked our car. Now, if you're young, you don't have this problem. You can drive into this lot and talk to your friends, and maybe the music is up. You go shopping for a few hours and you just walk straight out to your car. Your brain remembers where you parked it. And that's because our brains have this amazing capacity of forming what we call spatial maps. So that the brain uses cues in the environment, such as a lamppost or a storefront, to make a map in the brain that it can then instantly retrieve. But as we get older, it becomes harder to both to make these maps, but then also to retrieve them. And of course, in patients with Alzheimer's disease and dementia, this is particularly strongly affected or does not function at all anymore. It's interesting that animals have similar deficits when they get older. Dogs, for example, monkeys, but even mice, as you will see in a minute. So as we get older, our brain becomes more and more susceptible to cognitive dysfunction, neurodegeneration, and Alzheimer's disease. And the brain actually shrinks. It loses about 10% of its total volume from a healthy young person to a 90 or 100-year-old person. And it also loses some of the connection between the nerve cells in the brain that are essential for the brain to function and communicate between its different parts. And that's becoming an increasing problem. As you know, 32% of people above age 85 have Alzheimer's disease. And in fact, most of the people who have Alzheimer's disease are old or older. They are older than 75, 81% of them. And we have a total of more than 75 million people worldwide by 2030 is estimated. And that's really because people are getting older across the globe. This map here from the WHO shows in dark blue countries where you have by 2050 more than 30% or almost a third of the population will be older than 60. And in the lighter blue, 10 to 30% will be older than 60. But you see that this is really across the globe, people are getting older. And if you look at projections further out, you would have even more blue and people are getting older and older. So that generates a large population of people who become susceptible to these degenerative diseases of the brain. So what is aging? If aging is so crucial to diseases such as Alzheimer's disease, should we try to understand aging and maybe interfere with the process of aging itself? And this is really where the focus of my research has increasingly centered. And we're not the first one to think about this idea that we might be able to slow down aging or even reverse it. This is a 16th century painting from Lucas Cranach the Elder. And here you see people on the left-hand side, frail and old, enter this fountain of youth and they re-emerge on the other side, young and rejuvenated. So wouldn't it be great if that were possible, if we could indeed slow down the aspects of aging or even reverse it so that you wouldn't get these age-related diseases and you wouldn't get age-related dementia. And what I'm going to tell you now is at least in animals, this seems to be indeed possible. And I think over the coming years, decades, we may be able to achieve this even in humans. But let's first look at mice. So in experiments that were started a few years ago, now more than 10 years ago, actually, by a colleague of ours, Tom Rando, what he found is that if you treat old mice with young blood, either by infusing blood repeatedly or by surgically connecting a young and an old mouse such that their blood circulation is shared. Over several weeks of treatment and exposure to young blood, an old mouse becomes what we call rejuvenated. Many aspects of this mouse, as we'll see in the next few minutes, become more like a younger mouse. Many organs, including the brain, seem to benefit from this. And one experiment I just want to show you to really illustrate these powerful effects is what we call a parking lot test for mice. So this is actually called the Barnes maze after Carol Barnes, a neuroscientist. And it tests a similar aspect of memory and learning that you would use as a human when you park your car, you go shopping and you come back and try to find that car. And so in this case here, we have a big table that has lots of holes, and we put the mouse on this table, and the mouse wants to escape from this environment because it looks scary to it. It's open, it's bright and shiny, and they want to find a way to escape. And so we trained the mouse over a few days to find a single hole where we mount a tube underneath the table. So the mouse learns that there's a hole where it can escape into a dark, comfortable black tube. And all the other holes, it would fall to the ground, and it doesn't actually dare to go in there. It just sort of pokes its head in there, but doesn't go there. And the mouse uses the checkerboard here, the triangle and the rectangle, to make these spatial maps that I mentioned to you that humans also make. And so let's look how an old mouse performs on this. A young mouse would find this hole very quickly after we train it for a few times. But an old mouse, which here as a control was treated with a salt solution, this old mouse really can't remember where it parked its car or where it can escape from this table. And we trained them actually for 3 days, 4 times a day. And as you saw, this mouse cannot remember. But the next mouse I'm going to show you is the same age, a sibling of this mouse, but we treated it with small infusions of blood from a young mouse. So it got repeated infusions of young blood. Every 3 days, we give it a small infusion for a total of 3 weeks. And look how this mouse performs. So here, the mouse starts out, and again, it was trained the same number of times. And what you can see is almost the strategy is already different. And it walks more or less straight to that hole and escapes comfortably and fulfills that test. And if we do this many, many times over, we actually find that young plasma consistently improves the function of these mice. We find not only that memory is improved, but we can actually show that molecular functions, cellular functions are improved. So they have more stem cells and more stem cell activity, these mice. They have more activity between their neurons, what we call synaptic activity. Their neurons fire more, they have more electrical activity. And very important, as Dr. Andreasson said, with age, our brains become inflamed. The mouse brains become inflamed. And this young plasma can reduce brain inflammation. Now, how would this work? So how could something as simple as blood have these effects on the brain? The blood, of course, connects all the different tissues in our body. So it really samples from every tissue, but also delivers factors to every tissue. And so if we infuse this blood into the body, it will go everywhere. It will get to the brain. And in fact, what people found is that not only the brain benefits, and there's multiple references here that show in different labs that the brain benefits from exposure to young plasma or young blood, but also the muscle, as our colleague, Dr. Randolph, first showed, the liver, pancreas, heart, bone, aorta, kidney, even intervertebral disc seems to be regenerated and rejuvenated. So quite striking effects on these old mice here. And people have been able to identify some of the factors that are beneficial and that may mediate these effects. But I also discovered factors in old blood that seem to be bad. So it seems to be a combination that with age, you not only lose good factors in your blood, but you also start to accumulate bad factors. And some of these are related to inflammation. So we're actually not the only one with this blood rejuvenation that can show that aging can be slowed down or even be reversed. But probably one of the most amazing experiments that has been done in biology was conducted by Dr. Shinya Yamanaka, who actually got the Nobel Prize for his discoveries 8 years ago. And so basically what he can show, and this has now been reproduced hundreds of times and has even translated to the clinic for certain diseases, is that you can take skin cells from an old person or from an old animal. You can introduce 4 genes that are sort of critical switches in turning on a number of genes that can reprogram a cell such that the age of that cell is entirely lost, and you get a young cell that has essentially age 0 and can be reprogrammed to be a stem cell that can give rise to a new organism that is just as a young organism would be. Now, this is in cell culture, this is in a dish, but people have taken this to animals, again to mice, and can show, again, in an old mouse, if you introduce these factors and you turn them on and off sort of multiple times in a pulse-like fashion over a couple of weeks, you get a mouse that is again rejuvenated. And if people measure at the molecular level what is the age of this mouse, they can basically erase about 4 months of age in such a mouse. And that would amount in humans to probably 10 years or more. And similarly, in human fibroblasts, if you take that from a 90-year-old person, you can reprogram it to become much younger with this pulse-like activation of these factors. So here you don't make an embryonic cell out of it, but you just make the cell younger with this approach. So this suggests that indeed age, biological age of a cell or an organism is not something that is inevitable and is just progressing as it always does from what we know. But there are interventions, there are biological interventions that we know of today that suggest that aging is plastic and malleable, that can be changed. You can both accelerate it experimentally and you can reverse it. And this suggests then indeed that aging is a druggable target. And so there's now a number of companies that either directly try to target aspects of aging, and these are biotech companies here listed. Pharma is becoming interested. There's of course a whole area of dietary supplement or aesthetic procedures that try to slow down or reverse aging. And then there's lots of research that is going on in dedicated research institutions that are really trying to approach these age-related diseases such as dementia, Alzheimer's disease, by slowing down the aging process. But the big question is, of course, can this be translated to humans? And we actually started a company called Alcahes to really take that to the next level, because giving plasma or giving blood to people is something that we have done in the clinics for decades, and it's a very safe treatment. It has never been done systematically in patients with Alzheimer's disease, and certainly not by giving only plasma from young donors. So we conducted a couple of trials. Actually, the first one was done at Stanford by Dr. Sharon Shah at the ADRC. And these were proof of concept, sort of phase 1, phase 2 trials with small numbers of patients where they received multiple infusions of plasma from donors that are considerably younger than the patient. Usually the average donor age is about 35 years. And they clearly show that this treatment is safe. There's no unexpected adverse events. There were improvements in memory, but these were not blinded, not double-blinded, placebo-controlled trials yet. So we have to take this with a grain of salt, but they certainly look promising enough that this is worth continuing. So what's next in this area of research and potential treatment? Very encouraging phase 1, phase 2 experiments and trials suggest that it's warranted to conduct now large so-called phase 3 trials, where both the patient and the caregiver or the physician do not know whether the patient will get human plasma or just a salt solution. And then if we do a few hundred individuals with this treatment, we will find out whether this is really a useful treatment for slowing down Alzheimer's disease. We need to understand what the key ingredients are that are potentially beneficial in young plasma, and also the ones that are detrimental in old plasma. As I mentioned to you, as we get older, there's a lot of bad factors that accumulate. And so one way would be to simply inhibit these bad factors. And then based on that new knowledge, we would want to develop new therapies that target maybe these bad factors or simply use some of the good factors as a treatment. And so that's why we're so grateful that you are participating in these studies that we're doing as part of the ADRC. So we believe that as the brain gets older, and because the brain is part of the body and is so highly vascularized, so there's lots of blood that constantly pumps through our brains, that if you age and if you get a disease such as Alzheimer's, we will see this in the blood that many of you are donating, and we will be able to compare the blood from healthy people, from young people, or from age-matched old people who are healthy, and find out what is different. And look at this plasma. So there's lots and lots of things in plasma, lots of different proteins, and we can now measure thousands of these to find out what is different between a patient and a healthy control, and study how they change with disease to potentially then use these as new approaches for interventions. And so with that, I would like to stop here and thank you for your attention and be happy to take questions.
Thank you, Tony. So I just see for now only 2 questions, but Nicole, you might have picked up some more. I believe they're both right now directed. Looks okay. One's coming in soon for you, Tony. Just hold on. Let's start with Dr. Henderson. What typically causes the disparate results of the 2 tests that you were talking about? And I believe that has to do with the aducanumab question.
Well, no. So let me start off by saying that I did not, I was not an investigator in this trial, and there are a lot of details filed that have not been made publicly available, although some have. And for most studies to get FDA approval, the FDA requires 2 large pivotal trials that show safety and efficacy. And so the company making aducanumab conducted 2 parallel trials. The designs were similar. They took place in the US and also in other countries as well, because I think if the drug were successful, they would be looking for regulatory approval in Europe and Asia and so forth. The first of these trials did show a significant benefit of the treatment. Now, significant is a statistical term. It doesn't tell you how big the difference was. It just indicates that the difference was large enough that it probably wasn't due to chance by itself compared to placebo. The actual magnitude of that difference was really very small, but it was significant, and it was what the company set out to show, and they did show that. The other trial did not show this difference. It was completely negative, both in primary and secondary outcomes, at least my understanding is that. Looking then, picking through some of the subgroups within the second trial, they found some supportive evidence, and they thought that since Alzheimer's disease is a disorder with an unmet need, since one trial did show what they were aiming for, even though the second one didn't, then they would go to the FDA and seek approval for this. And we don't know what the FDA's final decision would be. One of the reasons for two trials is that sometimes things happen by chance. And investigators are different, patients are different, and even in the most controlled, most carefully defined settings, there can be differences. And it's not unusual for one trial to suggest something positive and a second and third trial to show different results that are a little bit different. So I hope that answers your question.
Okay.
I'm depending on you.
Yes, thank you, Victor. So there's one more for you, and then I believe there will be two. Well, I guess one more is on its way. And then we have
Why
some.
don't you go
Tony.
to the next question for Tony?
Okay, so we'll
For Tony.
alternate, I guess. And one of them went away, but I'll come back to that one. So Tony, should individuals with Parkinson's disease or other cognitive issues refrain from donating blood given your study results?
That's a great question. I don't know exactly what the guidance here is from the blood centers, but I think there are probably certain factors that are used to determine whether somebody can still give blood. I think at a certain age, people discourage from donating blood, but I wouldn't know exactly. I don't think there is. So the plasma that is given in these clinical trials is, as I said, from average donor age 35, although there are some individuals who are 60 or even older, but it's pooled. So it's a pool from hundreds or thousands of donors together. And so if one person were in there who were older, it would still give an average age of around 35, as I said.
And if I can add, I think and this isn't my area of expertise, but I think the primary concern with things like Alzheimer's disease and Parkinson's disease was whether there might be some transmissibility. There was some evidence in the laboratory that maybe some of the abnormal proteins might be able to transmit the disease. I think the conclusion has been that's not the case. And as Dr. Wyss-Coray said, that I don't think there's enough evidence that even if there are a few more of these deleterious factors or a few less, depending on the clinical population, that by itself, that would make an important clinical difference.
Right.
Okay, we'll come back to you again, Dr. Henderson. I guess we'll just go back and forth between the two. Dr. Henderson, are there any studies on the effects of exercise on dementia/Alzheimer's?
There are a lot of studies, and one of the wonderful things about research and one of the wonderful things about having a center like the ADRC is that, there are different perspectives on how to interpret some of the data. And I think my interpretation is a little bit more cautious than maybe some others. So there have been a lot of studies looking at people who exercise a lot and what, different kinds of outcomes, including dementia, including Alzheimer's disease. And most of them do show that people who engage in regular aerobic activity have a reduced chance of developing dementia and Alzheimer's disease later. The question is, who are the people who are doing the exercise? Are the people who are exercising the same people who eat well, watch what they eat, don't smoke cigarettes, have a healthy lifestyle, are aware of the potential importance of exercise, and are some of these other characteristics the factors that are associated with reduced risk, or is it the exercise itself? In animal models, it's pretty clear that exercise does a lot of good things with the brain, including reducing some of the chemical changes of Alzheimer's disease, but it's not as clear in people. And there is a large ongoing study in England, for example, that followed people over a period of 30 years or more. And the people who were exercising and want to develop dementia stopped exercising maybe 10 years before dementia was diagnosed, suggesting that maybe one of the early manifestations of cognitive loss is sort of an apathy. It's sort of a less of a tendency to want to become engaged and do these things. And so I'm actually fairly skeptical that exercise will have this major effect on dementia prevention, but I don't know. I think it's a really important question. We know exercise is really beneficial in a lot of other areas, and I certainly encourage my patients to exercise, but whether exercise by itself will make an important difference is unknown. And if I might add one other thing, there are several large multi-center trials, one in Finland, one in the US, several in other countries, looking at exercise plus diet, plus cardiovascular risk reduction, plus other factors to see whether or not in healthy people, this combination intensive intervention will reduce the risk of developing dementia. And the results aren't in. The Finnish study is the furthest along, and they did show after 2 years of analysis looking at cognition that there was an effect, a beneficial effect of this intensive intervention, but the benefit was very small, and it wouldn't be small enough to be individually noticed. It may be that the study hasn't gone on long enough, and maybe that for some reason this wasn't the optimal group to try to see the intervention. But I think I'm more enthusiastic now about the kind of research that our basic scientists are doing to come up with new ways of attacking this disease than I am about lifestyle interventions in and of themselves. But I'd love to be proven wrong.
Thank you.
I wonder whether Dr. Wyss-Coray wants to come in as well. I mean,
no, I think you made sort of the seminal points. In mice, we do see very striking effects, and people are starting to isolate individual factors now that are sufficient to replicate these effects. And so one idea is that these factors could then potentially be tested in humans. So again, the idea that you take individual components out of the blood and you turn them into medicines.
So if I might just reiterate what I think you're saying is that, plasma itself is sort of an intermediate tool on the way to what we hope would be a sort of a scientifically stronger answer. What are the individual factors that are contributing? And that would end up being, if they're identified, sort of the optimal treatment.
Yes, yeah, sorry, taking a step back. So studies have now shown that you can exercise a mouse and you can take the blood of that mouse and you can put it into a mouse that has not exercised, and you get the benefits on the brain, suggesting again that there's something that ends up in the blood, and the blood is sort of this carrier of information, very similar to when we take the young blood. There seems to be something in there that can be transferred into an old organism or a sick organism. So ideally, then you would get young blood from somebody who exercised. That might be the best.
So what we all need is a surrogate partner who will go to the gym for us.
Yeah.
So along similar lines, two questions having to do with diet with regards to the blood plasma. One is, have there been studies examining the blood plasma of older individuals who have followed the Mediterranean diet? And somewhat related in the diet phase is, how is fasting related to prolonged life, and is it related to any of these factors? Dr. Wyss-Coray and then Dr. Henderson.
Yeah, these are great questions. So what people are starting to do now is to measure thousands and thousands of different genes in brains of mice, as well as in other tissue, and see what changes when a brain gets old, and what changes in response to exercise, for example, or to diet, or to all these good interventions to giving young plasma. And what we find is that there are actually some common signatures, as we call them, as if there might be some commonalities between these interventions that have a benefit for the brain. And so again, the idea is then that we narrow down these signatures and find out what is it exactly that changes, and can we mimic that with a drug, with maybe a small molecule drug or with a protein drug that you could take as a pill. That's sort of the ultimate plan to do. But again, it's not clear yet what the key targets have to be to really achieve this with a significant effect. And it ultimately will have to be tested in humans, of course.
Thank you. We have a guest who would like to ask the question out loud, so I'm going to allow them to talk. Mr. Upsal, I hope I said your name correctly. I have allowed you to talk, so hopefully if you unmute, you can ask your question directly.
So can you see me now? Yeah.
Yeah.
There I am. Can you hear me?
Yes. Yes.
And my name is Richard Upsal, and my wife and I are both members of the Aging Brain Study. My wife, age 85, has been part of it for 2.5 years, and I've been part of it for almost 4 years.
And if I might break in there for the rest of you, the Healthy Aging Brain Study is in essence the same as the Alzheimer's Disease Research Center, but for historical reasons, sort of both names are attached to it. But go on, please.
Should I continue or give it up? You want me to continue?
Continue speaking, yes.
Okay, well, my wife is age 85 and I'm 88. So in the middle of September, my wife, age 85, had an extensive surgical procedure, which involved replacing 2 valves in her heart. They replaced a mechanical aortic valve and an organic mitral valve, both in the same procedure. And the procedure took quite a while. And at the end of the procedure, and a couple of days later, we noted, and even now, a significant change in her cognitive ability. She suffered a change in her ability to comprehend, and memory loss and so on, which as a result of the operation. And in talking to the physician who did the surgical work, he said, well, it could have been too long on the heart-lung machine, and the brain probably didn't get enough blood flow for a while, and it'll be something like a stroke. And I wonder if all of you understand that something like this can happen when you have a surgical procedure and you're an old person, and things are not so good for you mentally after it's done. The surgery took a long time because it was the replacement of a mechanical aortic valve, which is a big blob, and it took a big hole to get it out. And then they had to drag out an old valve, and it was a lot of work to do.
So what are you asking for us to comment? I think we
Yes,
would be.
I'm asking for you to comment. Go ahead.
Yeah, so if I might start, first of all, I'm sorry for what happened with your wife. I think long surgery is never easy on anyone. And as you say, it's more difficult on people when they're older. The question you ask is good because it's a question that comes up for a lot of other people. It's been known for a long time that people report, as you are, that after a surgical procedure, someone wasn't as good as before they went in. And then sometimes in going back, you can see that there were some changes going all along, and it wasn't just the surgery. But in other cases, that isn't clear. And there have been a number of studies now looking at people who've undergone anesthesia, who've been hospitalized, have had severe illnesses. And there is this linkage to being at risk for cognitive decline or dementia. It's not clear whether it's related to particular anesthetics, whether it's related to some other factor associated with being more severely, being sicker or something else. There are investigators at Stanford who are looking into this very issue, trying to understand the effects of surgery. And I think probably investigators elsewhere. And with regard to an actual stroke, an MRI brain scan would show whether or not that occurred. And that would be the answer in some cases, but often it's not that. And often we're sort of left with a bit of a mystery.
Yeah, I may be able to just give a sort of a biological perspective on that. A major surgery is like a major injury or wound in the body, and that leads to dramatic activation of the immune system. So the immune system goes into overdrive and alarm if there is this big cut to do the surgery, and that triggers all kinds of inflammatory pathways that could also be detrimental. So it's maybe a combination of different effects that just play out and have much more drastic consequences at old age than they would in a young person. And we're actually conducting such studies after major surgery to look at immune changes that accompany that.
Thank you, Dr. Henderson, Dr. Wyss-Coray. I hope that answered many people's questions. So I just want to let people know that we were going to take a break and I was going to lead you through some Qigong exercises, but I think we have a couple more questions that will probably take precedence over that. But if you need to take a break, feel free to do so now. I'll just continue with a couple more questions here, which have to do with more, I lost them. So coming back to MCI, what is the, how do we know about the genetics of different causes of MCI? And is there a role to combine traditional Chinese medicine with conventional therapy for MCI? That's for you, Dr. Henderson, I believe.
Oh, these combined questions. So thanks for asking. I think for what I tried to convey during my presentation is that conceptually MCI shares a lot with dementia. We think in most instances, some of the same changes related to plaques and tangles, for example, with Alzheimer's disease, are also occurring in MCI, but at a lower level. Some people are more resistant to these changes, have more resiliency, have more reserve to sort of not show the clinical manifestations. But I think the factors that would end up influencing the pathology of MCI would be the same as dementia. Nisha, so repeat that part of the question for me again.
I'm sorry, I was trying to scroll through the question. What was the second? Oh, about the Chinese medicine?
No, the first part of the question, whether there was an unanswered part to that.
Oh, the genetics with regards to.
So in terms of genetics, then it would be the same as for if we're looking at Alzheimer's disease or Parkinson's disease. And with rare exceptions, most of the genetic factors for disorders, these neurodegenerative disorders, are there, but they probably are operating at low levels. Things like ApoE is operating at a higher level for Alzheimer's disease and would operate at a higher level for MCI. But most of the other genetic factors are operating at a low level, and they would be the same as for Alzheimer's disease, for Parkinson's disease, for frontotemporal dementia, and so forth. With respect to traditional treatments and Chinese forms of medicine, there's been an increasing interest in this in the last 5 years, maybe 10 years, about effects these might have on cognition and dementia risk in general. We don't have a lot of clinical trial data there to show that they would be helpful. But again, we don't know for sure. So I think the answer there is it's an important area where we don't have sufficient answers yet.
Thank you. Dr. Wyss-Coray, a couple more for you. One is, how can people participate in these plasma experiments? And a second prong is, actually, these are all tied in. Is there reversed young blood becoming old? And then what was the difference between phase 1 and phase 2 in terms, sorry, not difference. How long did your phase 1 and phase 2 trials good effects last? 3 questions for you. How can people participate? The phase 1 and phase 2 trials, how long did the good effects last? And is there a reverse of young mice becoming old?
Yeah, great question. So the trials are clinical experimental trials. They were actually conducted, I believe, in Southern California. The second one, the first one was here at Stanford. There is no trial currently ongoing. So the company is planning how to conduct a large phase 3 trial. And so when that is initiated, then it would be, I think the easiest way is to find it on clinicaltrials.gov. I'm not sure if Stanford would participate in a larger trial, but it's not clear yet when that will be and how many participants. The question of whether an old or young mouse can get old, that's indeed the case. So we find when we give blood or plasma, the liquid fraction from blood, to a young mouse, so from an old mouse to a young mouse, we can actually accelerate many aspects of aging, and that mouse becomes cognitively impaired. So it shows similar signs to an old mouse, suggesting again that there are bad factors accumulating with age, and we have actually identified some of them that potentially could be neutralized to as a treatment for age-related cognitive impairment. And the third question was about
how long did the effects of
how long the effects last. Yeah, thank you. That's a bit harder to find out in humans, because usually when the trial ends, people are not participating in follow-up studies, at least not in this case, in these small trials. So I can't give you an answer in humans, but in mice, some of the effects last for quite a long time. We can measure changes in stem cells several months after the treatment was stopped, suggesting that it doesn't just go away after you stop the treatment. So that gives us some hope, but again, translation to humans is still pending or in progress.
Thank you. Another question, I guess, also related to the plasma is, oh, sorry, I lost it. It had to do with, have there been any studies done with cord blood, C-O-R-D? Maybe
Yes,
it's
so in mice, we have done that. So we try to get the youngest blood possible for experimental studies in mice. And there we actually find potentially even stronger effects, but practically it would not be possible in humans, because you would not be able to get enough cord plasma to treat a significant number of people, I think. There has been a study registered in Korea, at a hospital in Korea, but I've lost track of that study. They tried to use cord plasma to treat just age-related frailty in general.
Thank you. So I'm not sure if both of you can potentially address these, but there's a question about ketogenic diet and what patient profile it would be appropriate for. What is the role of diet in suppressing the anti-inflammatory effect? And if we get to it, there's a question about sugar and exercise and turmeric. Is it really anti-inflammatory? So maybe those first 2 questions. If either of you can answer them, if not, we'll just need to go.
I think
So what?
Before Victor.
I
Yeah.
think both of us could probably give partial answers. It's very clear that diet has an effect on inflammatory measures of inflammation in the blood. Some of this is related to the diet itself. Some of this is probably related to the microbiome, the kind of bacteria that a person has in his or her gut. But it does have the diet clearly has an effect on inflammatory markers. I think this is a really complicated field, and this is why I want Dr. Wyss-Coray to comment on this as well. But you know, the suggestion that inflammation has a key role in disorders like Alzheimer's disease goes back a long time. There have been clinical trials looking at anti-inflammatories that haven't shown a clear benefit in Alzheimer's disease. It doesn't mean that these are the definitive answers by any means, but the field is so complicated that I think, you know, looking at an anti-inflammatory diet is probably an overly simplistic way to do this. The ketogenic diet is a little bit different in that it sort of forces the brain to use a different form of energy. Instead of using sugars, it uses fatty acids. There's a lot of basic science to suggest that this would be beneficial to the brain for a lot of different reasons. The clinical evidence, like in other instances, isn't as strong as we would like it to be. And I think now, clinically, there's not specific recommendation to be made for this. There was a so-called medical food that was used for a while using medium chain fatty acids to try to induce a state of ketosis, which I think has been largely discredited, but other people are looking at similar kinds of approaches. Tony, would you like to comment as well?
No, I think you made the point. In animal studies, again, they look very promising, but they have not translated to humans.
Turmeric,
Sorry.
let me sort of jump to turmeric. It also has some anti-inflammatory effects. There have been clinical trials of this in Alzheimer's disease, which have been negative, but I would say the trials are really too small to show, to exclude the possibility of an effect, but the trials that have been done that I'm aware of haven't shown an effect.
And then what is the effect of sugar on for inflammation and neurodegenerative disease? And does exercise neutralize the effect of sugar in that regard, I'm assuming?
I think Dr. Wyss-Coray and I are sort of staring at ourselves through our through our Zoom monitor. Sugars do, like certain other foods, do have an effect on inflammatory markers like CRP, for example, in the blood. But there are a number of, you know, inflammation is not one concept. It isn't either there or not there. And as Dr. Andreasson in her presentation, I think Dr. Wyss-Coray has also suggested, there are a lot of different aspects to this. I don't think, and for a while people were more concerned about high fructose corn syrup compared to sucrose. And so the kind of sugar may make a difference. I don't know that those concerns have panned out clinically, but I think it's still an unanswered question. So whether or not sugars contribute importantly to neurodegeneration per se, I don't know. I think sugars do contribute to disorders like diabetes, which in turn are associated with both vascular disease, and I think also neurodegenerative changes like Alzheimer's disease, but I don't have a final answer. Exercise is beneficial on a number of different levels, but I won't try to speculate as to whether that benefit is mediated by the effects on blood glucose or sugars, because I don't know.
Thank you. Kristin will be talking about exercise shortly and PD, but I'm going to go ahead and ask this last question because that's, I think, hopefully going to wrap it up. And that is, what causes the plaques and tangles in the brain? That's a big question, actually. So what causes the plaques and tangles?
Let me sort of start with a sort of a broader overview, and then Dr. Wyss-Coray can sort of speak more directly to the biology. The plaques come before tangles in most instances in Alzheimer's disease. Plaques include abnormal nerve processes. Once they're better developed, they include amyloid in the center. There's a lot of evidence for inflammatory markers, activated microglia within the plaques themselves, and it's not completely clear what causes them. We do know that factors that increase amyloid genetically will increase the likelihood of developing these plaques. So genetic disorders, or the E4 allele is associated with more amyloid and associated with a greater tendency to develop these plaques. The tangles occur inside nerve cells, not between nerve cells like the plaques. They're made primarily of a form of tau protein, which is a normal supporting protein within nerve cells, but it has more phosphate groups than normal, which changes its binding capacity. These proteins also tend to fold abnormally, which can induce other proteins to fold abnormally. A lot of really good work has gone into trying to understand the link between the amyloid and the neurofibrillary tangles, their formation. And I think there are a lot of pathways by which this might occur. I'm not sure that it's really known exactly how it does occur. It's Tony?
Yeah, and unfortunately, we don't have the answers. Surprisingly, even though people have worked on this for 20 years, we know, as Dr. Henderson says, that there are genetic variants where you make more of A beta, for example. And we believe that is the cause of Alzheimer's disease in these patients. There is very good evidence that that's the case. But all of us make this A beta peptide all the time. And it seems even in patients with Alzheimer's disease, it's not that you make more, but it's that you cannot get rid of it. And Dr. Andreasson talked about this, that there are these immune cells in the brain, we call them microglia, that usually clear the A beta, they get rid of it. It's like the garbage truck is not driving through your neighborhood anymore. And so the A beta starts to accumulate. And interestingly, that accumulation then causes more inflammation in the brain. On the other hand, we also know that if you have an injury to the brain, you start to make more of the A beta. Then you're not just, if you're then old, you're not just failing to clear it, but you also make more of it. And so that may be particularly bad in people who have a trauma to their head. They have a fall, and it's similar to major surgeries that a lot of patients report that they had a fall and then it started to show problems with cognition. So it's still unclear why this peptide is produced in the first place, and that's why it may also not have such a big effect with these antibodies that then neutralize it, because it may not be the key or the only factor that is driving the disease.
Thank you. There's one last question, and we're going to have this be from a participant who will be speaking. So let me see if I can unmute you. Okay, Christina, well, hold on, here we go. Nami or Nomi, would you like to ask your question? And this will be the last one so we can move on with the other speakers. Nomi, you need to unmute.
Okay.
There we go.
I just wanted to say thank you. The presentations have been wonderful, very clear, very informative, and I thank the people working on this solution to this great problem. So it was just a thank you.
Thank you. Thank you for participating.
Wonderful. Thank you so much for that. If Kristin, if you could just bear with us, I'm going to ask everyone, if you're sitting, stand up off your chair for a moment and stretch your legs, and then we would we'll begin. And you might want to just raise your hands above your head, take a deep breath, and exhale. And do that 2 more times just to kind of inhale going up, exhale slowly coming down, bending at the knees. Now get a little taller, inhale going up, and exhale gently coming down. And then just kind of shake out your hands, shake out your feet, and take one last inhale going up with the hands and arms, and then gently exhale down. Now, this is not at all how I would typically do Qigong, but I just want to give you a quick break since you've been sitting for a while so that you can stretch and get that blood pumping all the way through up to the brain. So with that, thank you, Dr. Henderson, Dr. Wyss-Coray, and Dr. Andreasson for the first session. And next, I would like to introduce Kristin Morris. And Kristin, if you can just tell us a little bit about yourself as a self-introduction, that would be wonderful. Thank you.
Yeah, sure. I'm a physical therapist at the Stanford Neuroscience Health Center. I treat patients with all kinds of neurological disorders, as well as specializing in vestibular therapy for patients with dizziness. I've been at Stanford for 3 years, and before that came from way of Boston at Mass General and UCSF. Do you want me to start sharing my screen, Nusha?
Yes, please. Thank you so much for that
Yeah.
self-introduction.
All right, how do I look? All set?
Great. Yes, all good. Thank you.
Okay. So I'll be talking mostly about exercise for patients with Parkinson's, which can be applied to the broader spectrum of movement disorders, and also are, generally the same principles for patients with mild cognitive impairment or dementia. So just to briefly touch on learning objectives, as I said, I'll focus on exercise in patients with Parkinson's, and hopefully the participants will learn about the evidence to support exercise in this population, as well as how to implement some of these strategies at home and additional resources that may be available to you online. I really like this quote, so I'll read it out loud for all of you. Regular aerobic type exercise is the single strategy with compelling evidence for slowing Parkinson's disease progression. All patients with Parkinson's disease should be encouraged to engage in regular such exercise. So we know that exercise is very important. However, patients with Parkinson's typically spend 75% of all awake time in sedentary behaviors, such as sitting, eating, drinking, watching TV. Compared to healthy adults, this is 30% less activity overall. So the main take-home thing that I would want everyone to gain from this presentation is that exercise recommendations for patients with Parkinson's, which can again be expanded to other populations, are actually the same for all healthy adults, in that the American College of Sports Medicine and US Department of Health and Human Services recommend at least 150 minutes a week of moderate intensity aerobic exercise, as well as 2 days a week of strength training. For older adults, they recommend adding flexibility and balance training, which are also very important in people with Parkinson's disease. So now we're going to talk a little bit more about how do you know you're getting that moderate intensity, 150 minutes per week, which if you're doing the calculation in your head, that ends up to be about 30 minutes, 5 days a week of exercise. So one way to measure your exertion is subjectively. So this scale I put on the slide is called the rate of perceived exertion scale. And you can see 6 at the top is no exertion, that's just sitting and resting, whereas 20 at the bottom in the red zone, that's your maximal exertion. So that would be like a 30 seconds all out sprint. So where you want to be for sustained moderate intensity is around a 13 to 15, or somewhat hard to hard on this scale, which I've circled here in the yellow zone. Obviously, when you're first warming up, which everyone should always do a warm up for about 5 minutes, and then a cool down again, it's okay to be in this green zone when you're just getting started. A way to more objectively measure your heart rate and look at where your heart rate is in comparison to your exercise zones. So this is one app you can use to calculate your exercise zones. I like using this one. It's available in the Apple Store. It's called HR Zones. And I wanted to show you how to use the settings to make this most effective. So these are the only settings you have to change, and these are the only settings you can change. It's not too complicated. You want to toggle on this Carbonin formula, which factors in using your resting heart rate if you know it, as well as toggle on this calculation to calculate your maximum target heart rate. So that will give you the most accurate results. So once you've set that up, this is what the main screen looks like. And you input age, I put 65 as an example, and resting heart rate, I did 70 as an example. And this gives you a max heart rate of 161, and then shows you in percentages of that maximum heart rate, what your heart rate range would be. So let's start at the bottom here in healthy heart zone. This is great for beginners. So if you're new to aerobic exercise, if you can get up to this 50 to 60%, you're still getting some benefit of the aerobic exercise. Once you establish that you can build up your time there, I would encourage you to try to get into this yellow to orange zone, 60 to 80%, which is your moderate intensity aerobic exercise zone. Getting up into the orange and red here, it's not necessarily that it's unsafe. You know, of course, if you have any cardiac conditions, you'd want to consult with your doctor, or if you're on any cardiac medications that lower your heart rate response to exercise, such as beta blockers, it would get ranges specifically from a physician of where you want to be. But in general, it's okay if you get up into this 80 to 90% zone, but that's something you're only going to be able to hold for a short interval. We know that high intensity interval training is becoming much more popular and may have some added benefit for people with Parkinson's, but no definite research yet saying that high intensity intervals are better than sustained moderate intensity exercise. So first, in terms of aerobic exercise, I want to talk about walking. Simply put, if you want to get better at walking, you have to walk. Other forms of exercise will benefit walking. However, walking for exercise has the most benefit. If walking seems like it's limited by balance and that is impacting your ability to walk fast enough to get your heart rate up, you may need an assistive device. So this is where you'd consult with a physical therapist. A treadmill is also a good option for healthy adults or people with mild symptoms. I want to talk a little bit about this concept here of listening to a beat, which is called rhythmic auditory stimulation. Your typical walking beat or walking cadence is usually around 100 to 115 steps per minute. And especially in patients with Parkinson's, we see that if you hear a beat at that same rhythm, you'll naturally be inclined to walk to that beat without even thinking about it. And particularly with Parkinson's, this can give your brain a trigger cue for walking that bypasses the area of the brain in the basal ganglia that we know is impaired in automatically triggering a step. One way to figure out your walking cadence is to download this app called Metro Timer or any other metronome app. This one in particular has a button on it that makes it very easy where you can just, it says tap, and a family member or yourself, you can tap that button every time you take a step, and that'll give you your specific cadence. There's also some research that shows that listening to music at that cadence can have even better benefits for people who are musically inclined or feel like they can easily perceive a beat. Now, sometimes this can get distracting if you have greater cognitive impairment. So this is something that's particularly useful in earlier stages of the disease process. But once you find your rhythm, I would try listening to the metronome. So it's going to go beep, 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 and try that for a walk. And then if that seems to work well for you, you can go on YouTube or Spotify to look up songs. And I give some search terms here you would put in to find a song that matches your rhythm. And you may find that that's even more motivating and easier to walk to. So of course, there are other options for aerobic exercise other than walking. I put pictures of a few of them here. These pedalers over on the right, you can put in front of a chair and are good for people with mobility impairments. Moving up a level from that would be this recumbent elliptical in the top left corner, or recumbent stationary bike on the bottom here. And then if you have pretty good balance, can sit on a seat of an upright bike and could step over the bar of this upright stationary bike, that's a great option. I also included the elliptical and a rower, which are better for mild, maybe mild to moderate stages of disease. All right, moving on from aerobic exercise to strength training. We know that in Parkinson's disease, there are related reductions in muscle strength, and this can lead to difficulties with physical function, feeling less confident in balance, and increased risk for falls. The good news is that research supports that strength training can significantly improve muscle strength and performance of mobility. Most of these studies have been done in early to moderate stage disease, but that does not mean that they're not effective for those in later stages. It's just not as well studied. The important principle with strength training is that you need to always change your exercise or make it harder to make your muscles continue to adapt. So that means adding weight, adding repetitions, or switching up the type of exercise. I included here 6 basic exercises that target the primary muscle groups that we need for successful functional mobility. So I'll go over each of them with an example of an easier version and a harder version. The first one is a sit to stand exercise. So this is getting mainly your quads, some of your glutes, and you see the woman on the left here is using her hands to help herself push up, whereas the harder version on the right, you would do no hands and try to just tap that chair behind you and stand back up. On the bottom here, the hip bridge again gets the glutes and the back muscles. The easier version, the gentleman on the left is raising up with both legs. And then this harder version, you can try going on single leg only. The row muscle is a pulling motion that strengthens your upper back and shoulder muscles. So you can do a seated resistance band version as an easier exercise, or a standing one-arm row with a weight. And even if you don't have weights at home, you can use one of those reusable grocery bags we all have and put a few cans of food in there and you have weight. Moving down to the bottom here, hip abduction, that's your gluteus medius muscle, or a muscle on the side of your hips, which is very important for balance and walking. The version on the left here, the gentleman in the chair with the red band, he's pushing his knees out into that band and then slowly coming back in together. And the standing gentleman on the right is keeping nice and still in his pelvis and trunk and slowly bringing that leg out to the side and then lowering it back in. The last two here, we have the heel raise on top. So this is strengthening the calf muscles, which are very important for walking and balance. Easier version here on the left, raising up on 2 legs at a time with some hand support on the chair to push up if needed. And the harder version on the left, hands on the wall so you can't push up, and raising up on 1 leg only. Last exercise is a wall push-up as an easier version, and then harder version, a push-up on the floor on the knees. And there's obviously ranges of all of these, somewhere in the middle. For example, the wall push-up could be done on a counter to slightly increase the weight, then move to the floor on knees, and then obviously your standard push-up on your feet. So there's lots of creative ways to make these easier or more challenging. And this is just a small example of what you can do at home. I want to move on to mindfulness and yoga. There is a lot of attention now on exercise such as yoga, Tai Chi, Qigong, that all emphasize balance, strength, and incorporate this component of mindfulness. And I wanted to highlight this one study in particular that looked at participants engaging in a class that included mindfulness and yoga, and comparing that to another group who did exercise that included traditional stretching and strengthening. They found that there were additional benefits in the yoga and stretching group in terms of reducing psychological distress, improving spiritual well-being, and improving quality of life, while having the same benefits on motor symptoms and mobility that the traditional resistance training group had. In this study, the participants had one guided class per week that was 90 minutes, and then 2 20-minute sessions 2 times per week. So an example of how to implement this at home, Lauren will talk about later. We have some supportive care programs that would be great for your guided session, and you could learn a subsegment of that to do at home on your own. Other resources for mindfulness, as I mentioned, there are classes that combine balance, strength, and mindfulness through the supportive care program. Stanford Pain Medicine also has some videos that you can use on demand. So I put the link to that playlist here. It's a series called Tai Chi for Rehabilitation, and I would definitely recommend those as well. If you're looking to do your traditional strength training, but want to get the extra benefit of engaging in the mindfulness aspect, you can use apps like Headspace or Calm. And I additionally like these guided meditations from a physical therapist named Carolyn McManus online. And these are 10-minute guided meditations that one that's great. I really like the body scan that she does, and those are free and available at this link below. And I believe slides will be available for participants, so you can click on these hyperlinks later. All right, moving on to balance. So is there evidence that balance training improves balance for patients with Parkinson's? The answer is yes. I'll highlight 2 studies here. The first one looked at a multidimensional balance training program and looked at whether it improved gait performance in patients with Parkinson's. They found that it improved balance, walking speed, the ability to walk and dual tasks, meaning think about something else or do something else at the same time. And these improvements actually lasted for an entire year after the program was over. I believe that was an 8-week program. The second study down here, a highly challenging balance program, was found to reduce falls per month by 37%. These programs were both similar in that they were small group training that was done together and progressed by a physical therapist. So now we'll talk about how you can do balance training at home. One is through the classes through our supportive care program. Another is through other free online classes. So I put 2 links here. One is to the APDA, or American Parkinson's Disease Association website, and the other is to our Stanford Neurology page, which has a multitude of resources that have been put together since the pandemic started for free online classes, or some of them you can subscribe to. Another plug I'll put in is to ask for referrals to physical therapy so you can get an individualized balance training program. These are some ideas of what balance training at home might look like. I do want to mention that safety is always a concern. So when I have patients practice these at home, I usually want you to have ideally the corner of 2 walls behind you. So your back is to the apex of that corner, not leaning against the walls, but that way they're there if you lose your balance. And then it can also be helpful to have a chair next to you or in front of you, or a counter nearby if maybe your balance is better and you don't need the corner, but you just need something nearby. And then make sure there's no coffee tables or things to trip on. These pictures below just give you some ideas, like single leg balance, standing on one leg, walking, turning your head side to side is an example of dynamic balance training. And then closing eyes is an example of how you're retraining your body to use all of your different senses for balance. This one on the far left here, the clock taps, this one is fun at home. And you get an idea of this man at the bottom. He's stepping around the different numbers on the clock. So this app will randomly generate numbers. It'll say 1 o'clock, 3 o'clock, 7 o'clock. And you have to think in your mind, where is that number on the clock, and step to the target. And it has brain challenges as well, where it'll say months of the year instead, for example. So November would be 11 and you'd step to November. If you type in clock yourself to Google, you can watch a video on this, and the physical therapists who created this program also have an online balance training program that you can purchase. I have no relationship to them, but I use this app a lot with my patients. Do want to mention flexibility. In particular, patients with Parkinson's often end up with a forward flex posture, and the muscles that are in the front of the body often are the tightest, pulling into that posture. So some of these stretches help combat that tendency. We have the trunk twist laying on the floor, that's for your spine. The hip flexor stretch is for the muscle in the front of the hip. Hamstring stretch is for the muscle in the back of the thigh. The calf stretch here with the heel down on the ground, that's a really important one that's commonly gets tight. And then the chest stretch. So he's in the doorway with his hands on either side of the door, and you can do your hands up at different angles to get different parts of your chest with that one. So stretches should always be held at least 30 seconds at a time. You want to go into the stretch until you feel that not painful, but somewhat intense stretching sensation, and hold it there as long as it's comfortable. So I know that I talked about a lot of different forms of exercise, and people may be thinking, how do you fit this all in? So this is just one example of how you could structure your exercise for the week to ensure that you're getting all the components recommended by the American College of Sports Medicine. So you get your number of minutes for aerobic exercise. This incorporates some dance or balance exercise, resistance training, yoga and mindfulness, as well as flexibility. Just to touch on physical therapy, we do have our clinic. There's also other neuro clinics in the Bay Area. Home programs designed by physical therapists have been found to be as effective as going to a center and participating in the exercise only there, directly guided by someone. They showed significantly improved balance and walking speed and trends towards improving quality of life. This link down here is how you can find a physical therapist. You can select specialty, being neurology, to find a physical therapist in your area. This is sponsored by the American Physical Therapy Association. So this is a credible way to search for people who are certified in their neurology specialty. And then these are just some pictures of all the things that I like to do outside to stay active. This one in the middle on the bottom is actually me and my dad. He tried mountain biking with me for the first time in probably 40 years for him, right around his 70th birthday, and he had a great time. So never too late to start some of these things, as long as it's safe for you. And that's just references. All right.
Thank you so much, Kristin. A wonderful, informative presentation. Greatly appreciate it. And it was one that many people had asked for, so I hope it addresses, the interest that was there. Talisa, I think you have a question that you would like to.
Yeah, so a couple of people were asking what's the easiest way to get access to these exercises? I know you listed a few links. Do those directly tell you about the exercises you presented, and will they be able to print off those exercises for reference for themselves, or what's the best way to give them a copy of those exercises?
I can create a program online and email that out to participants, or I can give it to Nusha to email out to the participants. It's a little bit hard. I'll try to group it maybe into something for higher level and lower level, so that you're not getting confused with too many versions of the same exercise. And I had made a couple videos intending to be put on YouTube as a program that you could follow along, but we're still working to make sure with risk management and all these different departments that was okay. So if I get that up and running, I'll also forward that along.
That would be great. Thanks.
Great, thank you.
Just send it out to everybody that had. Well, I think I can just send it to everybody I have on the list. I think that was it, right, Talisa? I didn't see it. Well, looks like there's one more.
The last bio page. I'm not sure what that is. If you could show the last bio page, but I'm not sure what page that is.
Oh, the references, probably. Is that the page that we were looking for?
Not sure, but I do want to reiterate, as Kristin said, we will be making for both Kristin and Lauren have shared their slides, as has Dr. Henderson. So whoever's slides we get, we will be posting online on our website under the Participant Appreciation Day. Just kindly give us a few weeks to get that up by our webmaster, but we do plan to post the slides that were shared today. And hopefully we will even be able to post the recording. That is uncharted territory, but we'll do our best. So thank you for that, Kristin. I guess just leave it maybe another few seconds.
Misha, I wonder whether Kristin can make those references available to people who email you, whether you could forward them on her behalf or whether she could handle these directly.
I'm fine either way. Depends on Kristin's choice of how she'd like to handle that.
Yeah, I can, here, let me type my email up here so that everyone has it.
Because I think you said you were also going to put together those exercises into.
Yeah, and if anyone has any specific questions, not partners, I'm thinking of Boston, Stanford Healthcare. That's my email address, if anyone wants to email me directly, but we'll put everything together and email it out.
Thank you. Let's just leave that a moment and then we'll move on to Lauren. Oh, we have a raised hand. Okay, let me see if I can do this. Hold on one sec. Actually, it's not coming up. Okay. All right. Sorry. Yes, I'm going to let Brian Doherty ask a question. So unmute yourself, please.
Can you hear me now?
Okay.
Just a quick comment. I think the email is spelled wrong, isn't it?
Yes, I just saw that. Thank you.
That is all. Thank you.
Thank you.
Thank you.
Great. And if you think of more questions for the Q&A session, we'll handle them then. But I'd like to go ahead and move on to our next speaker. Thank you again, Kristin.
Yeah.
So our next speaker is Lauren Stroshane, and she coordinates the Neuroscience Supportive Care Program. I'll let her do a self-introduction as well. Lauren, are you on?
Yes, I am. Hi, can you hear me?
Thank you. Yes, please go ahead.
Great. So I will go ahead and share my screen. One moment. Okay, and is my screen visible to you?
Yes, but you probably want to go into the slideshow.
Yep.
There we go. Thank you.
Great. So I am so happy to be here. My name is Lauren Stroshane, and I'm the nurse coordinator with the Stanford Neuroscience Supportive Care Program. Our theme for today is fostering communities virtually through exercise workshops and support groups. I thought I would give a brief overview of the supportive care program and then walk you through the programs that we currently have available, explain what my role is as the nurse coordinator, and show you lastly how to contact us and how to actually register for any of our classes. So the supportive care program actually originated with the Stanford Cancer Center. So the cancer supportive care program has been around for many, many years and is quite well established. The neuro supportive care program is a little newer. It's just been around for a few years. And for, I think, almost a year, we didn't have a nurse in my current role. So a lot of what I've been doing now since I joined in August is trying to get the word out and let people know that we are still having classes and programs online during the COVID-19 pandemic, and that there's still a lot that we have to offer. Some of you may have seen some of our classes happening before in the Neuroscience Health Center down on the first floor in that big glass room, the Wellness Center. Currently, everything in person has been canceled for the time being due to safety concerns. But we are really happy that all our programs have been able to transition to Zoom, except for a couple. So the goal of the supportive care program really is to provide tools and support to all neuroscience patients and caregivers, and just to help promote the best quality of life possible for those living with and affected by these diseases. All our offerings are free, and you don't actually have to be a Stanford healthcare patient or caregiver of someone affiliated with Stanford in order to access them. They're available to the broader community, which is pretty neat. We have noticed since the start of the pandemic that for a lot of our participants, we get comments from them that it's been a surprisingly wonderful way for them to stay connected and feel a sense of community despite all the isolation that a lot of us are experiencing, needing to spend so much time at home right now. So that's been neat to hear. We also, to be honest, it's a very small silver lining of the pandemic that it forced us to transition to online. This means a lot of our programs have become accessible to people who wouldn't otherwise have ever been able to come in person, whether they live very far away. We have some participants from out of state even, or whether they just have a lot of transportation challenges that prevent them from coming in person. So I'm going to walk you through our current offerings. Over here on the right, we have our virtual flyer. I know it's small to read. Really, I just wanted people to see what our flyer looks like. In case you see it floating around in clinic at some point, that's what it looks like. So our programs really fall into three main categories: exercise classes, other types of classes and workshops, and then support groups. And I'll talk about each in some more detail. So Kristin Morris did a wonderful job just now of making the case for why exercise is so important for people with neurologic disorders. I mean, really for all of us, but especially those with a diagnosis. Some of our offerings are geared towards anyone, just a general neuro population. Some are a little more disease specific, and I'll talk about that. Those that are really open to anyone include our most popular program, which is Tai Chi. That happens, I think, twice a week and is really popular. We are very excited to offer, starting this coming week, we are opening up Tai Chi in Mandarin and English as part of our ongoing efforts to try to include more language variety in our offerings. So we're really excited to be starting Mandarin in English and as well. We also have Qigong, which our wonderful Nusha Asghari, our moderator today, is the instructor for that, and that's been fantastic, and Yoga for Strength. Our middle category are Parkinson's specific. That said, we generally encourage people with the Parkinson's diagnosis to be the ones to participate. At the same time, we do allow for people who have maybe a related diagnosis such as Lewy body dementia or essential tremor or other diagnoses to still join. So if you or someone that you know doesn't have Parkinson's disease but is really interested to try Dance for Parkinson's, for example, which is one of our most popular classes, you're welcome to reach out to us. We can talk with you, assess what you're looking for, and make sure that that's appropriate for you, and then you're welcome to participate. We don't want to turn anyone away. And then our last category are offerings that you certainly you can have normal mobility and participate in these, but they're particularly geared towards the needs of those with significant mobility limitations. So someone who is maybe in a wheelchair or has a lot of balance issues, anything where you're worried about falling, those with a tendency to have drops in blood pressure. We often encourage to participate in these classes. In pretty much all of our offerings, you're going to be experiencing likely mild to moderate exercise, according to what Kristin had described. None of these are going to be, super intense boot camp style, CrossFit types of classes. And the instructors all, are experienced and have expertise in working with individuals affected by neurologic disease. We have a number of classes and workshops. I thought I would highlight 3 here today. And then you're welcome to visit our website for a more complete list. We recently opened up mindfulness meditation for the neuro side of supportive care. This is one of our few classes that's available both within cancer supportive care and neuro. So it's a mix of people who attend. But it's proven really popular as a great way for folks to, find ways to de-stress and just gain some more skills around mindfulness and being present. And that's available 3 days a week as well. Healing Strokes is one of our newer programs that's been really popular. It's an art therapy based class for stroke survivors and their caregivers. And again, still free, but we do actually mail the art supplies to the participants' home so that they can participate. And that's actually led by some Stanford students who came up with the idea. It's been really neat to see their enthusiasm. Lastly, I wanted to highlight a series of workshops that's coming in January 2021 that is specifically for the caregivers of those with memory disorders. That's going to be probably about 8 workshops of 90 minutes or so each, covering a range of different topics, such as resilience and bouncing back, strategies for managing stress and dealing with challenging behavior, coping with frustration, that sort of thing. One thing that is neat about that workshop that we haven't done before is you can just register for whichever sessions are most interesting. You don't need to commit to a 90-minute session for 8 weeks, which can be really challenging for caregivers. So we're aiming to build that as a more flexible program so that more people can access it. And lastly, I wanted to mention the support groups and support platform that we offer. We have a memory disorders caregiver phone support group that meets once a month. So for those who are caring for a loved one with mild cognitive impairment, Alzheimer's disease, Lewy body dementia, or other types of dementia, it can connect with others via the phone, and just it can be a really wonderful way to get support, ideas, learn about other resources that are out there in the community, and just generally, feel less alone. We also have a phone support group for those affected by Huntington's disease, which meets quarterly, so less often. And then a really lively, well-attended discussion group for those living with multiple sclerosis that meets every week. Our last offering currently is the Smart Patients online community. It's an online platform for those affected by stroke. And it's basically a forum where people can post questions, post resources, advice, that kind of thing. So it's not as structured or doesn't require as much time commitment as you know, a regular support group. So for those who have been affected by stroke, we have that partnership between Smart Patients and Stanford. So I wanted to finish out my presentation by kind of walking you through our website a little bit, just to show you what it looks like and how to register, since I know that can be a little bit of a hurdle for some. Up here at the top is our web address, stanfordhealthcare.org/NSCP. So that's the best place to learn more and to register for any of our classes. Over here on the left is what our homepage looks like at the top. And if you scroll down, you can see the list of our current offerings. This is updated every week, so it's very current. We don't generally have anything that's outdated on there. If you scroll down the page, you'll see a list of the different categories that I was just describing. So our classes and workshops, exercise and fitness and support groups, and you can click on those headers to see the lists of the actual classes. When you click on one of those classes, you'll get a pop-up that looks like this, that describes, for example, chair yoga, gives you a little more information about what to expect and how you can prepare, what you would need, for instance. All of our classes are really designed for someone who's working out at home. So, you know, none of our classes are expecting you to have a fancy Peloton or some, you know, very specific piece of equipment. I think the only thing you might need is a sturdy chair, maybe a pad. If you have some free weights, depending on the class, that can be helpful. But, you know, our goal is to make it as accessible and low stress as possible. So you'll see a description, what you would need, and then event details. So when it happens and how to register, which takes me to our next slide. So let's say that you find something that you want to register for, and you know, you're interested to see it on the calendar and sign up. You'll scroll back up to the top of our webpage and click on this gray or black, depending on your monitor, button that says class calendar. And that's going to pop up a long text that is our participant agreement, which is about safety and confidentiality while you're participating with other individuals in the classes. If you agree to that, then you can click to proceed to our calendar, which has all the lists of all the different days of our classes, and you can click on those classes to register. So there are a lot of reasons why people might want to contact us directly. Maybe your neurologist told you, you know, the importance of exercise. Your physical therapist, Kristin, just told you, you know, how important exercise is, and you know that, you know, it's something that you want to try, but you're not really sure which exercise class or which workshop might be a good fit for you. You might also want to know what are other resources and services that are available to me through Stanford or other organizations in the community. And we can help with both of those questions. So you can, if you want to just learn more about the classes and register, as I mentioned before, stanfordhealthcare.org/NSCP is our website, but you can also reach out to us by phone. 650-721-8500 is our general line. If you leave us a voicemail on that number, either I will call you or my colleague, Graham Wellman, who is our program coordinator and handles a lot of the logistical elements elements of our programs. You can also email us at nscp@stanfordhealthcare.org. And again, one of us will respond to you that way as well. And the last option would be maybe having one of your providers, whether it's your neurologist or your physical therapist or social worker, can also put in a referral to supportive care, which just triggers me to reach out to you either way. And either route is fine. We don't have a preference, but I am available, to do one-on-one consultations with you to help you figure out, what exercise class or support group or workshop might fit your goals and your abilities. And then I can also talk with you if you have other needs that aren't being met and see if there are other resources or services that might be helpful. And with that closes out my presentation. So I'm happy to answer questions now.
Thank you so much, Lauren. Talisa, there were a couple of questions. Would you like
Yes.
to go ahead and moderate that?
Yeah, so there were about 3 questions, I think. So the first question, would the classes be available for specifically a younger patient population, specifically someone diagnosed with POTS? If you're not sure what that is, for all the participants, this is also known as postural orthostatic tachycardia syndrome.
Yeah, absolutely. Yeah, any neurologic diagnosis would be welcome. For someone with POTS, I'm somewhat familiar with it, so I know that there can sometimes be concerns. Sorry, I don't know. I think my screen just stopped sharing for some reason. But let me know if I should put try to share again so that the contact information is available. Yeah, anyway, someone with POTS, depending on how severe their symptoms are, I might need to speak with them and just see if there are modifications that they should do to participate safely. If it's an upright class, I might recommend having a sturdy chair on hand, making sure that their environment has somewhere that they can sit or lay down quickly if they start to feel lightheaded, just as an example. But yes, you would be welcome to participate.
Okay, great, thank you. And the second question is, we also have a participant that is a Lewy body dementia support group facilitator. And she was just wondering if she had permission to share these resources with her support group members to give them that information.
Yeah, absolutely. Please do. Feel free to reach out to me if you need any more information or our virtual brochure, for instance. But yeah, the more the merrier. Thank you for asking.
And then lastly, the last question is just asking, are these classes that you provide free, or is there a charge for participants or patients?
Yeah, so all our offerings are free. There is only one exception, which is one of the workshops, not one of the ones that I highlighted today. It's called 8 Steps to a Healthier You. But everything else, all our exercise classes are free, whether you are a Stanford patient or not.
Great, thank you. I think that was all of the questions that I had come up for you, unless there's anything else. Oh, I did have one question just came in. Are the classes, are they available online, just kind of at your own pace, or do you take them at a scheduled time?
Yeah, that's a great question. So they are at set times, and you can go on our website and see our calendar that lists, on Tuesdays we have this, and this at these times. It is over Zoom. So what happens is that you'll register on our website. You're not charged anything. It's just so that we have information to contact you. And then you'll receive a Zoom link by email. When it's time for the class, you'll click on that link to log in. But yes, it is at specific times. And unfortunately, because of privacy concerns, we don't record any of our programs currently. So it's not something that you can watch later.
Great, thank you so much for that information.
Yeah, you're very welcome.
And I think at this point, we should just take maybe another 5 minutes in terms of any questions for both Kristin and Lauren in terms of anything that they've presented. Thank you both so very much, Kristin and Lauren. Those were wonderful, informative presentations. And while we wait to see if any questions come through, since I didn't get to put this plug in earlier, I'll go ahead and put in a plug that the Qigong class is actually, Qigong is a Chinese medical health exercise. It actually integrates mindfulness. I'm actually both a yoga instructor and a Qigong and Tai Chi instructor, so all three of those. So I integrate all three of those into the Qigong class, which is held Thursdays at 3 p.m. and is sponsored by the ADRC. So it's a good mix of all of those. Similarly, the slides, again, we will do our best to get them up as soon as we can of those that we've received from the speakers. I'll reach out to all of them again to see what we can post, and we'll try and have that within the next few weeks. So with that, are there any additional questions for either Kristin or Lauren at this time? And if you'd like to speak, please raise your hand. Otherwise, please type it in the Q&A. Although
I thought of one thing I didn't mention earlier, which I meant to, which is just that in order to participate in any of the Zoom classes, you will need, just like you are today using Zoom, you will need a computer, an internet connection and a camera and a microphone. You're not required to keep your camera on if you prefer some privacy. A lot of other participants do tend to keep their cameras on just so that they can see each other and interact a little bit, but it's not required.
Thank you. That was helpful, actually. Thank you for sharing that. So I'm not, oh, let's see, one chat. Oh, that's very kind. Okay. Wonderful. Thank you for that. Yeah, I hope you've all benefited from today's event. And Christina Wyss-Coray had a few things to share. If there are no more questions for either Lauren or Kristin, we can move into our closing phase. I just want to say thank you to everybody again. All of our speakers, thank you so much. As well as all of our participants, thank you for joining. So Christina, would you like to share a few, you had a few closing remarks before Dr. Henderson closes the event.
Yeah, thank you so much. I also want to thank all the speakers. There were great talks, and thank you to all the participants logging in today. I just have a couple of things I wanted to bring up. So we have started a newsletter. Hopefully you all got that about a month ago, and we are now doing every quarterly, we are planning a newsletter from the ADRC, and we will in the future, update new trials that are coming out, and also like, I had a lot of ideas of today's presentation, what we can add to the next newsletter. Then whoever participated today, we will get a survey out by email. It will be towards the end of next week. And this is just a feedback of this event today. We like to hear what you think and what was good, but also what we can improve on. So if you would fill out that survey for us, that would be great. It's a very short one, just should take a couple of minutes. But then we have another survey that we would like to send out to the ADRC and UDAL participants. And this is a COVID-related survey, and just to get some input on the effects that COVID had on you. It's a little longer. It should probably takes 5 to 10 minutes to fill this out, but we would appreciate if you could do that. We will send that by email as well, and we will be available to follow up by phone if there's any questions. That's all what I have today. Again, thank you so much for participating, and I will hand over to Dr. Henderson for the final words. Thanks. Bye.
Thank you. I have no prepared final words, but there are a couple things I want to say. First of all, the COVID survey is one of the things that's being collected by the National Institute on Aging. All the Alzheimer's centers are asked to do this. And so it's a way of trying to let people know on a national level how COVID has affected people enrolled in these kind of research projects, whether you've fared fairly well or haven't. I'm sure everyone's impacted by the social limitations. So we appreciate your doing that. And before I sort of thank everyone else again, let me point out the difficulties with COVID and how the effects it has had on clinical research, like the Alzheimer's Disease Research Center, like the Pacific Udall Center. Different centers in different parts of the country are able to deal with this in different ways, primarily because of local laws and regulations. Here in our part of California, Santa Clara County, people are being particularly conservative about what we can do and what we can't do. Stanford University is very concerned about how patients and research participants and staff and faculty might be impacted by this pandemic. But I think we're finally starting to see our way clear to begin and resume some of the clinical evaluations that we weren't able to do before. There are new procedures and safeguards being put in place to make sure that it can be done safely. I want to thank everyone within our centers who's been participating remotely, doing follow-ups by telephone and by video. Thank you for doing that. We hope that some of you, or perhaps most of you, will be able to re-engage in person if we can see that that can be done safely. And we hope to begin enrolling more people the way we originally said we were going to do during this funding cycle. And it's been largely put on hold, but I think now we can also start that process carefully and cautiously. And Kristina is the person who is sort of keeping the closest track in all this, and the one who has the most responsibility for making sure that it goes off without a hitch. So one more thing that she has in her plate of things to do, but we look forward to being able to continue in our mission within the centers. I thank Nusha today for organizing this conference. I think it's really a large undertaking to get speakers together to figure out the logistics and manage all of us. And so thank you, Nusha, for that. I thank our coordinators for monitoring the questions that came in about connectivity. I hope that for most of you, it worked out all right. And as always, I thank people here for joining in. I hope you learned something interesting and new, something that you can think about a bit, and maybe some things that you can do or activities that you might want to become engaged in. In the surveys that you answered that Kristina mentioned, let us know what you might like to hear next year, things that you're particularly interested in, things you want to know more about. Let us know how we're doing. Once again, thank you for joining us, and enjoy the rest of the afternoon. Do so safely, and hopefully 2020 will be ending in a way that is happier and safer for all of us. Thank you.